Thank you so much, Jason. And thank you all for organizing this wonderful event. It's really an honor to be here today and to be able to share my work with you all today. So the title of my work is Visualizing Some of Nature's Most Mysterious Molecular Machines using cryo-EM. And this protagonist of the short story that I'll tell you all today is YME1L. And so here's a close-up of this intermitochondrial membrane protease inside the mitochondria really at work and starting to chew up misfolded proteins to maintain mitochondrial quality control and health. So throughout my entire career, a central thesis that has really inspired my work, whether I was looking at microtubule binding proteins that stabilize this cytoskeletal protein, or viral entry proteins that can hijack our machinery in order to do their work, or this protein that I'll focus on today, YME1L, this protease, I have found that structure provides great insight into an enzyme's role within the cell. So mitochondria are double membrane organelles, and that means they're isolated from the cytosolic ubiquitin proteasome quality control system. And so what I've found very beautiful is that they have been able to evolve this team, or this army, if you will, of proteases to maintain quality control and health. And one that really stuck out to me and inspired my pursuit of this postdoctoral project was YME1L. So it's this intermitochondrial protease located in the inner membrane of the mitochondria. It's ATP dependent. It's a hexamer of ATPases as well as proteases. And what really stuck out to me is that it's highly versatile. Not only does it perform processive degradation and eat up misfolded proteins and toxic proteins within the mitochondria, but it can also perform site-specific cleavage, pull apart huge aggregates, as well as chaperone proteins across the membrane. So I was really impressed with this versatility, as well as this decision-making. So how does it know what to do and when, and what does that mean in disease? We see that when there are mutations in YME1L that affect its ability to processively degrade, in mice under stress, specifically a high-fat diet, they die immediately. But in cancer cells, we see that when YME1L is knocked out, there is a decrease in cell growth, an increase in apoptosis, and it serves almost as a tumor suppressor. But when there are mutations that affect its site-specific cleavage and affect proteins that balance mitochondrial dynamics and fission and fusion events, we see in patients that chemoresistance can be increased as well as malignant progression. So there's this versatility to how it's sensing and responding to stress and then changing how it chooses different and targets different substrates and processes them. So I was very impressed by this stress sensing capability of this protease. And so in 2017, the Lander lab resolved the structure of YME1L, starting to translocate different substrates. So what you're looking at here are the ring of ATPases and the ring of proteases. And what's happening is the ATPases are engaging a protein of interest that it wants to process and really intercalating like a zipper along the backbone of this protein and pulling it into its degradation chamber to be processed. However, this was really only one snapshot of the protein in action. So when I joined the lab, I had questions about substrate recognition. How is it engaging substrates? How is it releasing substrates? How are these other potential conformational states, significant conformational states along its landscape? What role are they playing in these critical decision-making processes? How does this change under stress and what type of stress? Is it tissue specific? And how does it change in the eye versus the heart or the brain? And how does age affect all of these decisions? Lots of very exciting and interesting questions. So how do we get from sample to structure? So I can do a bacterial expression and pull out the catalytic domain of YME1. Then I can freeze it and get it ready for it to process it using cryo-electron microscopy. I plunge freeze the sample. This happens very fast, 100 meters per second. And then I can image it using a high-end microscope that has a beam that moves at 1/3 the speed of light, paired with a very high-end camera to take images of the protein in action. And what really stood out to me as soon as I could get 2D images of YME1, specifically the yeast active homolog of this protein, was the motion. So here you're looking at the substrate translocating state, and it looks almost like a stop sign. But what I want you to notice is the other states where it looks like that stop sign is almost split in half and is completely moving in these dramatic conformational rearrangements. It's doing these conformational gymnastics. And so what turned out to be really critical was using programs that are motion-based in order to sort these out. So I found 3 states that were critical along its trajectory, or most popular. And one was the open state, so that split hexamer state. Then there was this potentially capture or release state. And then finally, the canonical closed state. And these started to give insight into how the protein was potentially engaging substrate. And using motion-based neural network models, after a lot of hard work, I was able to get high resolution images of each of these states. And what made me so excited is I could see that this split state was significant, was a real state along the entire conformational landscape of this protein. You're seeing the ATPases split apart. Here's a side view, so you can see how split the hexamer is. It's really opening wide open. And then you have the protease domains that were also splitting open. And this was really surprising given how dramatically covalent and tight these interactions are. So to see them pull apart, that was very dramatic and unexpected. And what was nice is we could use these states to now collect or to put together an entire story. And so I'll show you a short movie that illustrates that. So first, I want you to imagine that YME1 is in the mitochondria, it's probed and engaged and ready to start to eat substrates or process substrates. And so here in orange is a substrate. You can imagine it's like a little Cheeto and it's hungry, it's ready to eat that. And so it's primed and open. And then what we see is that it starts to position and potentially read the fold or read the sequence of the substrate before positioning it into the central pore and starting to commit or decide, how should this be processed? Site-specifically cleaved or processively degraded or chaperoned? What should I do? Before making a commitment finally to degrade it and to completely chew it up, essentially making a commitment to destruction. And so the takeaway message that I really want all of you to have today is that we have now the full conformational landscape of YME1. We also have biochemical data that nicely complements that this state is off. And so it's off, it hasn't performed cleavage yet. And what we're seeing is that in the presence of substrate, upon different nucleotide rearrangements or different processes, you have these conformational rearrangements that allow it to start to engage and chew up its protein of interest. And so that was my main postdoctoral work. It was very focused on the yeast catalytic domain of YME1. And what makes me excited moving forward are these same conformational gymnastics and these processes, are they also observed within the human? And what does that mean for drug development, identifying potential pockets that would be great for new therapeutic development? What can we learn from this process to understand more about substrate specificity? Is it sequence specific? Is it fold specific? And in some combinations or in some situations, is it a combination of both? And how does YME1L sense fission and fusion dynamics or enzymes that drive mitochondrial dynamics? And what does that mean for the overall health of the protein or of the entire cell? And so I'm very excited to think about looking at the full-length protein and really building upon what I've done in cryo-EM, but now looking at the proteins in situ, within the cell. And that's really where I would love to see these proteins in action, building and working with each other to modulate mitochondrial crosstalk and meet specifically high energy demands within different tissues. And I love the idea of comparing high energy tissues like in the eye with both healthy and disease states to really understand the mechanisms at play and think about rational therapeutic design moving forward. And with that, I'm at time, and I would love to thank my amazing colleagues, my postdoctoral mentors, PhD mentors, and all the mentors who, out of their own heart, are able to give their time and their effort towards my journey as a future faculty individual. And of course, all of you for your attention. And thank you all so much.